This is the introduction to lecture 14, which will just start us off on looking at plant development. And we'll talk a little bit about how plants differ from animals in general terms, and we'll look at all the different processes and so on that take place. But I will also point out a similarity. And then, well, many similarities, really. And then we'll just, in this one, we'll look a little bit at the life cycle of Arabidopsis, which is the main model organism for this field. There are others. And we'll just look really briefly, introduce axis development. The axes are not the same as in animals. Some fate maps. You can begin at very early stages of embryos. And these are some of the earlier to later stages of development. And then we'll talk just really briefly about if you were to slice, through one of these embryos, you would see that the cells on the outside were different from some of the ones further in and others in the middle. And so there's multiple layers, and these are radial structures because they are circular. And the idea is, how do you shape this? How do you make it grow? And there's different ways this occurs through changing the plane of cell division, and that's a main theme for that kind of growth. And I'm trying to remember, but I think we will probably talk about it again a bit later in the section on plants. And then whether cell lineage or position is most important. There's very little movement of plant cells. In fact, there's pretty much none unless you do it artificially. So this is a weird question to ask, but you can ask it and you can answer it through experimental manipulation. And then I'll just really briefly introduce auxin, which is, there are a number of plant hormones that you've probably heard about if you've taken the appropriate courses, but auxin is the main one that we'll discuss for this class. And it has many different roles in development at different times. And we'll talk about directional transport and that sort of thing next time. Okay, so just a few basic terms. I'm sure you already know these, but above the ground we have the shoot, and this part is referred to as the shoot, this part as the root, of course. I'm not really that concerned if you remember, root hairs and tip and all that, but just to know, we aren't probably going to spend time on the patterning and development of the root, but the processes are somewhat similar to what we see above the ground. So we're staying above ground for the most part. And so if you were to look inside, this is where we see the radial symmetry with the epidermis on the outside, and then the connective tissue, and not connective, the conductive tissue sort of in the center, and then a layer around here, which we'll look at in a minute. So that's where the radial symmetry is, and then this is the apical part of the plant. Apical means away from the ground, and basal means towards the ground. So this is the basal side of the plant. And so this structure is more basal than this one. This structure is more apical than this one, and so on. So we're looking at this. If this was anterior posterior, then just change it to apical and basal. It's still polarity, right? And then of course the organs. When we talk about the organs of a plant, we're talking, and they are referred to that way in publications sometimes. We're talking about the leaves and in some cases the flowers. And then the apical bud is the one right at the tip. And if you've ever had a house plant, you may know that if you're trying to get a house plant to grow more bushy, you sometimes just pinch off the apical bud, because then what happens is the lateral buds, which are in between, so the nodes are where the leaves emerge, and then right above, exactly where the leaf emerges, there's a little bud in there, and that has the ability to form a shoot and to continue on the growth. But in many plants, it's dormant. But once you remove the apical bud, then some of these lateral buds will sort of take over, and then you can get these sort of bushier plants. The internode region is the space between one node and the other on the same side of the plant, and the same over here. This would be the internode region on this side, and so on. And then the fruit. We're not going to talk about fruiting, except that that's where the seeds are. And that's where development kind of begins, is in those seeds. Okay, so how are plants different from animals? Plants are not mobile, of course, as you already know, but I'm pointing it out because what it means is that plants have to be somewhat flexible, because whatever environmental conditions occur, they can speed up and slow down development. Day length determines when plants flower and that sort of thing. So there's sometimes strong environmental changes influencing development, and it's in many cases the environmental influence on plants is stronger. Some of the organs are formed post embryonically from special tissues that are set aside, called meristems, and the meristem tissues are capable of continuing to generate new leaves, new shoots, sets of flowers, and so on, and new roots, and branches on roots, and so on, even well into the adult stage of a plant. You can have a plant 500 years old, a tree or something, and it still has meristems, and it could still form properly new flowers, new leaves, and that sort of thing. Cells within a plant are surrounded by a cell wall. It's quite inflexible. It's sturdy for support and so on. But what this means is that changing the shape of something, or even growing certain structures in a particular direction, will depend on sort of differential growth. So more growth in one area means that part of the plant grows larger, but also if you change the plane of cell division, a tissue can get wider rather than taller, and so on. And we'll talk about two kinds of cell division today. And there's lots of developmental plasticity. Some of you, if you've grown houseplants at all, or you have family members who do, you may know that for like a plant like an African violet, sometimes if a leaf is removed from the plant and then stuck into some soil, the little leaf can grow a whole new entire plant. And there was no root there. The leaf is able to, where it was cut from the plant, those cells can change and start forming roots and so on. If you've ever taken a cutting from a plant and put it in water and had the roots start to grow and so on, this is called developmental plasticity, and the ability to regenerate, because you can, cut off the inflorescence, the floral stem of a plant, and it can make more, such as this orchid here. This is one of my orchids. I took the opportunity to put a picture of it. But when this was growing, I bumped into it and I knocked the flower stem off with the flowers. And so, that was a real nuisance. So I put it in water to keep the flower. But then another little shoot came up, and here's, some more flowers from this orchid. So they're able to regenerate things when they get damaged and so on. So the mechanisms of development sometimes look different, but the aims of development are pretty much the same. So the processes, an axis is set up. There's polarity. One end of the organism is different from the other. Cells differentiate to form different structures. There is cell signaling, so cells can talk to, communicate with each other, send information. There is position-dependent differentiation, so a cell will take on a different fate depending on its position, what signals it's receiving from the surrounding cells. There are transcription factors that act together or interact with others to turn on and off certain sets of genes that sets the cell fate. And there is also cell-cell communication, not just through the channels in the cell walls called plasmodesmata, but there are also some molecules that are able to slip through the cell wall. We always think of the cell wall as this kind of thick wall. When you hear the word wall, you think of like the walls of a building or something. But they're actually more like a meshwork. And so there are small molecules that are able to move through the cell wall from one cell to another. So there is lots of communication. So the strategies and the aims are the same. You know, you want to make sure each cell knows where it is in the organism and what it's supposed to be forming, and so on. The difference is, though, that the genes are different, and the actual way you get there is somewhat different. For example, there's no such thing as gastrulation in a plant because the cells, due to having that cell wall, are not able to migrate, move around. Instead of migration and moving around, you have these differential cell growth, so more growth in one direction than another to create these shape changes. Arabidopsis is a small and pretty easy to grow dicot plant, and it's also diploid, and that's a very useful thing. Many plants are polyploid, which means they have maybe in some cases up to 6 copies of every gene. You cannot do good genetics very easily with an organism that has 6 copies of, because most mutations are recessive. You would have to make sure all 6 copies were mutated in order to see the phenotype. You can imagine that's really difficult. It has a almost completely sequenced genome, and it has 27,000 genes, so a few thousand more than you do. And so yeah, but it's still a relatively small genome. So there's not, corn has massive amounts of I can't remember, 35,000 genes or something. So Arabidopsis, relatively speaking, is simpler. So gametogenesis occurs in the flowers, of course. There are stamens and carpel. The carpel is the female part. The stamens are the male parts, and we'll talk about fertilization briefly in a minute. And basically, pollen is produced and ovules that are inside the stigma of the plant, the female part. And the pollen basically lands on the stigma, and then the tube, a pollen tube, grows right through that and makes its way through and into where the ovules are, and it basically delivers the sperm nuclei there. And that creates, for each of these ovules that gets properly fertilized, a little zygote, and then the embryo develops. And we're going to spend time today talking about these different structures. And as that embryo is forming, this shows you one of the stages, which is the globular stage. And this is the mature stage. It's almost a seedling. So we're spending lots of time talking about the different processes here. And then that leads us to germination. And the germination, when we look at that seedling, the roots, of course, are below the ground. And the shoot is above the ground, but the stem up to where the leaves first start appearing. These are cotyledons. They're not real leaves, but but after that, from this shoot, the leaves are going to form. So this is the hypocotyl. So it's below. Hypo means below, and then the cotyledons. And then from here, the shoots and the leaves and so on are going to develop, and so on. And that might be enough for now, because we're going to look at some of these stages in more detail momentarily. So just point out pollination. Pollination is really interesting, and I was going to talk about plant fertilization, but I decided not to because it's quite complicated. Yeah, it's quite complicated, and there's not a lot of good resources in the textbook that describe it in detail. So I thought I would just explain it a little bit here. So this is the stigma of the pistil, which is the female part of the plant. And the anthers are on the stamens. And basically, some plants self-pollinate and some have mechanisms to prevent self-pollination. So there's a lot of signaling and recognition that goes on here. First of all, the key things is you want to make sure that you're being pollinated by the correct species of pollen. So when you look at pollen grains, they're highly diverse in how they are structured. They have often these little spikes and funny shapes and so on. And Dr. Rolf Matthews in our department is an absolute expert on fossil pollens. He can see little fossils of pollen and know what kind of plant basically produced that, thousands and thousands of years ago, millions probably. Oops. And so, for heaven's sake. And so in any case, so there's recognition that happens here. And then if it's the correct species, the pollen basically sends out a long tube. This is all still one cell, and it extends all the way down until it gets inside where the ovariole where the ovules are. So each of these is an ovule, and it contains not just the egg, but other structures, which I don't want to talk about too much. And then 2 sperm nuclei are sent down here, and they make their way in here. Now, one of the sperm nuclei fertilizes the egg, so now we have a diploid. And it's complicated, but these 2 other nuclei from the female plus a sperm nucleus together makes the endosperm, which is triploid, 3N. And it's important only because it's the source of nutrition. As this embryo is growing, the only way it can feed is by the starch that is provided by the endosperm. So that's a nutritional, it's like yolk in an egg. It provides nutrition for the growing embryo. Okay, and then the embryo grows in there until it reaches the seedling stage, and then, then the process completes. Okay, so the egg is fertilized and it forms the zygote. And then the very first division that occurs is called the 2-cell stage, and there's a larger cell on the bottom and a smaller cell on the top. And the top one, the little one, is going to be the apical cell, and the bottom one is called the basal cell. And then the apical cell will divide to form the octant stage. So there's you can only see 4, but on the other side there are 4 more. So there's 8 cells in there, which is why it's called octant. So a couple of divisions have taken place here. And then the basal cell has divided too, but it is divided in a linear fashion with the planes of division this way, so that you have this long stalk that's kind of holding things up. The basal cell continues to be specified as basal, and this tissue keeps dividing to form the suspensor. The suspensor is a supportive structure. Once the mature embryo is fully grown, the suspensor gets crushed and obliviated. But it is the source of nutrition for the growing embryo and support for the structure. Yeah, and so the triangular stage is, when further development has taken place, and it's just based on, the apparent appearance here. By the heart stage, there are certain tissues that are set aside in different places that are kind of like a fate map. Okay. The torpedo stage, the cotyledons, which are going to be those first 2 seed leaves that also provide nutrition until the embryo till the seedling starts growing its own shoots and leaves and photosynthesizing and so on. And so it's referred to as the torpedo stage, but I don't know who named it, because it's quite obviously the rabbit stage. I mean, I don't know what people were thinking. But in any case, like, whatever you do to remember this, we can remember. And this, when I hear the word torpedo, that doesn't help me remember this stage at all, because I don't think of a torpedo like this. But I do think of a rabbit like this, and this helps me remember. Okay, and then the mature embryo, the cotyledons are huge, and then the rest of the structure here, the vascular tissue is already forming. You've got ground tissue, which is part of the inside of the radial symmetry, with the epidermal cells on the outside, and you're going to have presumptive root and so on in this region. So that's a brief run through that early development. And the apical, or sorry, the seedling has, as I said, the apical and basal polarity, as well as the radial polarity. So the epidermis is on the outside, and all the vascular tissue is in the center, and then this cortex is the region in between. And as the plant grows, the stem can get wider, and then we'll talk later about sort of if that stem is going to get wider, how are the cell divisions organized. Seedling structures, you can actually make fate maps if you do cell marking and following and that sort of thing. You can actually see the cell fates. And by the time we get to the torpedo stage, but as early as the heart stage, most of the tissues have been specified, but not all. So the heart, not quite, but by the torpedo stage, yes. And so what we have is there's a presumptive root and then root meristem tissue. So meristem tissue is the tissue that gives rise to further roots. So as this root is starting to grow, there will be lateral ones being sent to one side or the other. Think of, your basic root, but then, when you look, there's sort of branches off the roots and stuff. And so that's going to come from here. The hypocaudal is going to be this part here. That's the part that's sort of between right here. And then there's this little part, which is sort of the connection between the hypocaudal and then the actual cotyledons, which will be the leaves eventually. And this little bit on the between the ears of the bunny, that's the shoot meristem. So the growing shoot is going to come out of here. Leaves are going to form from here. And this tissue is self-renewing, so it stays the same size all the time, with some cells differentiating and forming new leaves and stems and that sort of thing. So the torpedo stage is really important because that's the point at which we really know all the tissues that are going to give rise to the adult structures eventually have been specified at this point. Now the radial axis is established by the globular stage, and this is a schematic, and this is what it looks like under the microscope with the suspensor, the basal cell down here, and then if we were to take a slice through here and then turn it so that you could be looking straight down from here, looking down on it, then you would see C here, which is again the vascular tissue, this bit here, and the cortex, which is this bit here, and these two together are referred to as the ground tissue. They're conductive tissue and supportive tissue, and then the epidermis is the layer on the outside. It's like the skin of the plant, if you like. Now, how do we shape this structure? There are two different types of cell divisions. There are periclinal. Peri means beside or around, and so basically that's the wrong color to use. Basically what happens, so you've got these cells and they're forming this ring, and what you want is you want to form an additional ring, right? You want the ring to be wider, 2 cells thick instead of 1 cell thick. And so what you do is when cell division is happening, you basically, the plane of division is oriented so that one of the cells is close to the inner layer and the other one's on the outside. And so now you have, when all of the cells have finally done this, then now you have an additional layer on the outside. And so it's 2 layers thick, right? But when you make another layer on the outside layer is going to be a little bit bigger than the inside layer because it's a wider thing. And so you have to add some additional cells into that ring to make it larger. And so some of the cells divide in what we call anticlinal. Anti is against, right? And so periclinal is beside or in the same plane as the actual ring, and anticlinal is in the opposite, perpendicular to the actual ring. And so for periclinal, we can make more rings when we divide that way. And then as the plant is growing, the rings, if you have additional rings, they have to get wider. And so the rings get wider when the cells divide this way. And there's other kinds of structures that can be formed using this kind of strategy. And I'm trying to remember if we talk about them or not. I'll check the lectures and let you know. Okay. So that's one thing. The next question, which is just part of the introduction, is to just ask if plant cell fate is due to cell lineage, that is, the different cell divisions it's gone through, what its parent cell was, what genes it happens to be expressing, and so on, or position. This is kind of like asking if the cells are specified or determined in a way. And so a cell that is primarily, it's sort of, this question is weird in a way, right? Because it sort of doesn't matter, because cells don't change position in plant development. But what this tells you really is it the genetic history? Is it the changes in cell fate over time, historically, that determine what the cell is going to become? Or is it generally signaling and communication? Because if it's signaling and communication, then when you move a cell to a new position, then it will actually change its fate because it's receiving signals and information from the surrounding cells. But if it's due to cell lineage and it's already permanently shut off certain genes so it can only form X structure, then moving it is not going to cause it to change its fate. And so there's two different kinds of methods you can use. One is an experimental method where you can ablate parts of a tissue and then observe what happens to the cells that move into that empty space. And the cells don't really move there, so we kind of have to help them with some micro manipulations. But if when you do that manipulation, the cells do change their fate, then that means it is the position that matters most. Whereas we can also take a genetic approach. If you were to disrupt the regular pattern of cell division, so the cells divide in a certain way, and they form a very orderly structure with cells at one end taking on one fate and cells in the other end taking on another fate, then that suggests that they've had these genetic changes that have occurred through the history of those cell divisions, certain genes turned on, certain genes turned off. And so if you mess up the pattern of cell division and have them, kind of random cell divisions and one cell dividing way more times and the planes of division being all messed up, then you would expect, if cell lineage is what really matters, you expect you'd have this horrifyingly just chaotic embryo, right? So lineage would matter in this case. This would be the thing. So here's the results, and I'm going to go over them in the class. But so there are two, the ablation one. So if you ablate some tissue with a laser or something like that, and then you kind of force them into a new position, we find that the cells will develop according to their new position, not their lineage. And a genetic experiment, there are some mutations called FAS, and there's a few others. But the cells, they don't divide along the planes that they should be. They're dividing with random sort of plane of division. And they do form intact plants. with all the tissues present, although they look strange, but they do have, a stem. They have an apical versus a basal. They have radial symmetry. They have leafy structures that look kind of funny because of the weird cell division, but the leaves are there and they're specified correctly. And they even have flowers, which also look kind of strange. So they're misshapen because of the cell division, but the actual types of tissue that are formed are correct. Okay, so think about that, and because I'll probably ask a question on it in the class. Okay, so the first division is that asymmetric division, and the AB, apical basal polarity is established. I've already kind of talked about this stuff, so this is just to remind you. And it turns out that auxin, gradients of indole acetic acid, which is auxin, is the most important signaling molecule in many of these early developmental processes. It does different jobs in different parts of the plant, and we'll talk about some of those in probably the next lecture. Okay, and it promotes the changes in gene expression by its effect on proteins that are called OXIAA, ox-indole acetic acid proteins, and these happen to be repressors of auxin responsive factors that actually transcribe the genes, what we call the auxin responsive genes. So we treat with auxin, and certain genes are transcribed, and they're transcribed by these ARFs, auxin responsive factors. But the mechanism that is not as direct as you would expect. Okay, and I'll talk about that in 1 second to show you just kind of how that works. Right after the first division, we've got these two cells, and what happens is auxin is transported up into the apical cell, and as further divisions occur, auxin stays up in the apical region and can be produced in the lower cells, but it gets transported up into the apical cell. And it's needed to specify apical fate. So if you mess with any of the auxin signaling, you mess up the apical basal fate. Okay, so how auxin works. There are auxin responsive genes, and they are transcribed by auxin responsive factors. And there's lots of these because auxin is involved in many different processes. So if there are different response factors, being expressed, they will turn on a different subset of the genes that are transcribed based on auxin signaling. But there are these OXIAA proteins, and they are busy stopping the ARFs from doing the transcription of those genes. So here comes auxin into the picture. Auxin comes in, it binds to and interacts with these proteins, and it basically targets them for destruction. So you may remember from cell biology classes that ubiquitin tagging is how proteins are targeted for proteasome degradation. So sometimes they're phosphorylated first. I don't think that's the case here. I think they just get a whole bunch of ubiquitin molecules added to them, and they get sent to the proteasome, which is kind of like the garbage can of the cell. And proteins that go in there get degraded into amino acids so that those amino acids can be recycled and reused. And so because these guys are inactivated, now the ARF proteins are perfectly able to bind and initiate transcription of the target genes. So auxins, it has the role of promoting transcription, but it does it in this weird backwards way, because what it's really doing is stopping the repressor from doing the repression and allowing the ARF factors to get on with the job of transcribing those target genes. Okay, so in the next lecture, we'll talk a little bit more about, just a little bit about what auxin does, a bit about how it was discovered and how it helps pattern the plant embryo. And we'll spend time talking about directional transport, because a key to auxin activity in plants and its effect on development is it being transported in different directions, and especially when we talk about in a couple of lectures sort of where leaves are placed. there's this directional rush of auxin in one direction, and then it moves into another direction, and it all gets sent in another place, and then a leaf develops, and then the auxin gets sent somewhere else, and so on. It's this very interesting process. Okay, we will see you in class.